on the NF. Thank you, Jose. So you are on the NF-Core channel for the special interest group on animal genomics. And so this channel brings together a crowd of people who are interested in genomics analysis on farm animal with a special interest on NF-Core and Nextflow and related pipelines. And this is how this all started. But of course, the talks can be more generic, and we are typically looking into forward-looking talks where we look at what is going on, for instance, in human genomics and on the frontiers of human genomics. And we try to alternate between this and between talks that are truly centered on animal farm genomics. And today we are very happy to welcome the first speaker of this year. I have to warn you that the meeting will be recorded. So if you talk or if you show your image, this may appear on the recordings. So you may want to know about this. And so I'm very, very happy to welcome Ted Kalbfleisch. I hope I'm pronouncing it right, Ted. And Ted is a professor at the University of Kentucky in the Gluck Equine Research Center. Ted is actually a physical chemist. He received his PhD in '94 from Boston University. He describes himself as a bioinformatician, although I think he's much more than this and has done a lot of work in genetics, in bioinformatics, in protein-protein interaction. One of the most relevant aspects to this channel is that he led the development effort of the EQCAB3 reference genome for horse, which I understand was the first T2T assembly, and I believe this is one of the things he will tell us about today. His work is really focused on building infrastructures to support genetics and genomics research on horses, and I don't have to tell you how fascinating a model system horse is, since it is both a stock animal and a pet, and so it has real extraordinary available data associated with, and that's something we'll be very interested to hear about today. Now, you're welcome to ask questions on the Slack and on the chat channel, and we will keep an eye and ask questions on your behalf if you want. But at the end of the talk, there will be a question and answer session chaired by Francesca Bernini, who is helping me chairing this. Thank you very much. And Ted, the floor is yours. Thank you again for coming today. This is great. We're already at 45 participants. This is amazing. I think we're going to break our record.
Oh, goodness. Now I have to figure out how to make this go small. There we go. Thank you very much, Cedric, for that very kind introduction. I'm very pleased to be here, and I'm excited to talk to you all today. I hope very much you find it interesting. Okay, so the title of my talk today, The Post-Genome Era. How did we get here and where are we going? I am at the University of Kentucky, very fortunate to be there, and I saw in the gallery any number of horse people. It is wonderful to see you, as well as all the other species that you good folks represent. Thank you for attending. Okay, so there we go. So I would ask the question, what have we accomplished in the last 29 years? So I'll speak a bit about the first 14 of it. They were a golden age, I believe, for metaphors and memes. We were drinking from the fire hose. We were changing tires on moving cars. If you were a bioinformaticist and you were giving a presentation, it was almost law that you had to have this graph, which was the growth of data within the sequence read archive at NCBI. Note that this is logarithmic, and it hasn't slowed down since 2021. People depositing high quality data to this public resource that can be mined by those who have the interest in doing it. It was an exciting time to work in this domain. I consider myself very lucky to be a part of all of that. We were drunk with enthusiasm, but there were some things that we had not enough of. We didn't have enough computing power. We didn't have enough bandwidth. So back in the day when you needed to move data, it was not at all unreasonable to unplug a hard drive, put it on a plane, and send it halfway across the country if you needed to move terabytes of data. Terabytes of data were a very big thing back then. A terabyte of data back in 2000 cost $1 million. Right, I saw somebody write a check for $17 million to buy 17 terabytes of storage. Right, you can just walk down to whatever your local electronics store is these days, and for 20 or so bucks, you can buy a thumb drive that has terabytes of storage on it. Just to put this in context, I am old enough to have gone into a computer store to buy a computer, and I was speaking with the salesman. How much storage do I need? I was thinking, and he said, this one comes with a 10 megabyte hard drive. He pauses, and then he says, you will never generate 10 megabytes of data. Well, there are those of us on the call who have probably sent 10 megabyte emails, right? So yeah, that's something that's in our rearview mirror. One thing that again, we didn't have good standards based software, expertise programmers and database developers, or money. But that was the very beginning. But those things radically changed. What about them changed? Okay. So let's look at this larger timeline. In 1988, the Human Genome Project was initiated. In 1995, microarray technology showed up, and at that point we were genome wide. Right, you could put assays for tens of thousands of single nucleotide variants, of transcriptome probes, whatever, right? You could put those on a chip and you could ask a question of the whole in a relatively unbiased fashion. What is going on in this particular sample, right? What are its genotypes? What are its transcription values? And that introduced a change to science. So science prior to that had always been hypothesis testing. Right? Then we moved into a generation where it was legitimate to think about hypothesis generation. So we moved into a new era in science, and again, that was exciting to be a part of. In 2003, the human reference genome was completed for the first time. By 2007, next generation sequencing emerged with the 454 platform, and in 2009, a genome for a human being was published, and that cost $1 million. If you go all the way back here to 1988 through 2003, that work took 15 years, cost $2.3 billion, all the king's horses and all the king's men. This work, which led to the publication of a finished genome, or rather of a sequenced genome that could be mapped back to the human genome, that could be done for $1 million, right? So 2.3 million down to $1 million in 2007. In 2008, RNA-seq burst onto the scene, and we could begin to do truly unbiased work. Both the next generation sequencing technologies, both with respect to whole genome shotgun sequencing and RNA-seq, made the analysis of genomes truly unbiased. If it was in the tube, you got sequence data for it, right? This was a game changer for us, and our genomes began to show their seams a bit. We were getting a lot of reads that didn't map back to the genome. Well, they had to map back to something. What did that mean? Well, it meant that there was chunks that there were chunks of our genomes that were missing. Either because the individual had content that wasn't in the reference genome, or the reference genome was just a bit shy. It could have been either of those things. Then things changed in a big way again in 2018 with ONT reads. We could begin to sequence very, very long sequence data, and they and PacBio got us past these repeats that were nightmarish hellscapes if you were trying to assemble a genome. So now that you had these data, you could contemplate the notion of actually assembling something that went through really repeat-rich regions like centromeres and into telomeric regions and so forth, right? So the notion that we could assemble a genome in its entirety was born here. And around 2022, a telomere-to-telomere genome was published. And for the last, for the last time, I think that somebody could say we have the first fully complete genome for a human being, and that was a great thing. PacBio Revio showed up, and the price of these long reads went through the floor. So you went from $2.3 billion and 15 years down to a project that could be done for about $20,000 in reagent costs by students or postdocs in my lab in a semester. If you want to drive the price of anything through the floor, give it to the biotech industry, or it would seem speed it up. What did we do in all of this time? We built genomes. Phasing was a real problem for us early on, but we were okay with that. We cataloged variation in species and between individuals. We learned in detail and largely without bias how transcriptomes changed, tissue to tissue, cell type to cell type, and condition to condition. In here, this was an era where short read data dominated. And again, just to go back to price and how we've blown through every perceived barrier. Once upon a time, there was this thing called the X Prize. It was going to give $1 million to somebody who was able to sequence a whole human genome for under $1,000. Right. So I'm buying. I'm having some sequencing done, and it's costing me, I don't know, $2,000, and I send samples out. I pay the bill, and then the next time I go to send out samples, the price is down to about $600. We blew through that $1,000 $1,000 barrier and didn't even notice it. Things have changed, and in almost all ways, in very good ways. So for me, the next big change in genome sequencing happened when a buddy of mine, Tim Smith, had a great idea. So previously, when one sequenced when one went to build a genome, so phasing was a problem, right? You're sequencing a diploid animal, and what you're ultimately building is a haploid representation of that individual, right? So you got a maternal chromosome, you got a paternal chromosome. They're producing content. Sometimes it doesn't agree. You gotta fight through all of that. So the strategy had been, and the rat and the mouse people, they could, they were in a great place, right? They had animals that were practically haploid. They were so inbred. And they had been working on this for 200 generations. Dr. Ansack, who's here on the meeting today, the horse that was used for the ECCAB3 assembly, Twilight, she was considered inbred for a thoroughbred, right? And she still had a polymorphism about once every 100 or so bases. So when assembling it, that still made for problems. Tim had this clever idea that, since we had these ultra long reads that were going to have a lot of information about alleles that were specific to that animal, what if instead of looking for animals that were really, really inbred, look for ones that were really, really outbred? Right. So the idea, and in BOSS, this was a fairly easy animal to find. And Dr. Peterson, she was able to come with a an F1 hybrid that was a cross between a yak and a Highland bull, and a bright young fellow by the name of Ed Rice took the hi-fi data that was produced for this animal, right, and the hi-fi reads that came off the machine. Right, so they were 10 to 15 kb long, and they contained enough information, and the parents differed enough genetically that you could identify the parent of origin of each read. You could bin it separately. You could bin the maternal reads. You could bin the paternal reads, and you could independently assemble them and get haplotype resolved assemblies for the maternal and the paternal chromosomes for this calf. So those were submitted, and there was a period of time, I don't know exactly how long it was, but the best genome in the whole world belonged to a yak. So that F1 business, we in the horse world can play that game quite well too. And again, Cornell, Dr. Anzac's herd, we were able to find a mule who had a thoroughbred dam and a donkey sire. We sequenced that animal, and typically what had been done to do this, the trio sequencing, is you would sequence the sire and you would sequence the dam. You would take those data sets and you would generate k-mers to find animal-specific k-mers. So alleles that belong specifically to the sire, alleles that belong specifically to the dam, and you would use those to identify the reads and split them up. Okay, the PacBio hi-fi data got really good in the time between when Ed Rice, Jessica Peterson, and Tim Smith did that yak assembly, and when we did our mule assembly. So we generated the short read data on the mule, on the dam and the donkey sire. We assembled the hi-fi data, and we got 2.5 gigabases of donkey sequence with a contag N50 of 35.6, which back in the day was pretty good, and a thoroughbred assembly at 2.6 gigabytes. And this was just a contag level assembly at 43 an N50 of 43.5, all pretty good. But then I asked myself, okay, so we're guiding this software with the maternal and the paternal DNA. What if I don't have maternal and paternal DNA? This is going to be a problem for a lot of people who obtain rare samples. We took the F1 data for this animal, we pushed it through the same assembly software with no parental data at all. The PacBio hi-fi data was so accurate that the assembler was able to recognize that reads for orthologous regions in these chromosomes did not belong together, and they assembled them independently anyway. So just working with this animal's hi-fi data, we got 5.2 gigabases of sequence, which coincidentally is about 2.6 plus 2.5, and a contag N50 of 38.3. So the data were good enough that phasing and assembly, maternal and paternal, was certainly possible with F1s, and was conceivably possible with interspecies, with intraspecies crosses. Forgive me. Okay, then came ultra long read assembly, and they precipitated what ultimately became T to T assemblies. So what has been wonderful, and I haven't spoken in depth to the people at these companies at all, but it has been fantastic that they have each carved out their niches. And PacBio did the really accurate mid-range stuff between 13 and 20 kb, and the PacBio reads, or in the ONT reads rather, just kept getting longer and longer and longer. because we needed both of those. We needed both of those to be able to get across those repeat-rich regions. The ONT data, they gave you the contiguity. They could get you across the centromeres. The PacBio data had enough accuracy that when combined with the ONT data could accurately translate what was on those ultra long reads into a very accurate representation of these regions that had previously been incredibly difficult to sequence. So we can routinely generate 25x or more of reads that are greater than 100 kb. How is this not magic? My lab, with samples that we've sequenced, has seen 3 megabase reads. 3 megabases. Holy cow. So I spoke with somebody, with Miten Jain, who has a deep knowledge of this technology, and I asked him the question, what's the longest read that you can produce? What's the upper limit? And his answer was, there isn't one. In principle, if the molecule can fit into the pore, it can be sequenced. So if the biophysics allows, there's no reason that we shouldn't ultimately be able to get chromosome length reads. Wow. Okay, so our current abilities to build haplotype phase genome. Now, this is not, I don't want to pitch this as a completely solved problem, suggesting that there's no button that one can push that is going to be able to produce an assembly. But in the hands of experts who know what they're doing, a fully phased telomere-to-telomere genome is something that's possible. So what we produced for the thoroughbred was initially had telomeres at both ends of all chromosomes but 6. And with the help of Eleonora Capoletti, who's on the meeting today, we went back into the data and we dug around and we found that what the problem was, that even with the ultra-long data, even with the hi-fi data, we weren't able to get through the centromeres on these acrocentric chromosomes. And the centromeres that, so each of these things would have been missing about, anywhere between 5 and maybe 13 megabases of sequence. What we had to do then is we had to go in and we had to mask those centromeric regions. And when we did, then with the proximity ligation data, that's also a part of this equation that I haven't talked about much, but with that data, we were able to assign these hitherto unincorporated contigs onto the ends of these chromosomes that had been missing their telomeres and fill the gaps that they left. So currently we've got 5 gaps that remain, 1 on chromosome 16, 3 on chromosome 19, and 1 on chromosome 27. So all pretty good. The mule that we used to build this telomere-to-telomere assembly, we harvested 141 tissues from hers. We have 9 samples of each of them. These things are in cold storage at Texas A&M. So one thing that we learned in the FANG project was that we had a modest amount of money. And we were able to sequence maybe 8 or so tissues from the horses that we had collected for that study. Yet we had banked many tissues. What was initiated in the FANG effort that was led by Dr. Jessica Peterson, Dr. Carrie Finot, and Rebecca Ballone was an adopt a tissue effort. Right? So if you came to us with your credit card and you said, I'm really interested in what's going on in that tissue, you could pay to have that tissue sequenced. And by doing that, we were able to backfill with a whole lot more tissues than we would have been able to sequence with the funding that we had been able to collect. So this was a wonderful thing, and we're doing it again with these mule tissues. We've done isoseq on 14 tissues, but we've got 141 that are available for IsoSeq as well, or epigenetic ChIP-seq, any other sequencing technology that would be useful to doing further annotation on this animal. These tissues are available. So the horse T reference genome annotation, right? This is where the rubber meets the road. You got the genome, but if you don't know what's on it, what good is it? We've got 4 teams that are currently working to annotate these. One team is constructing a pangenome with the T as a foundation. I'm going to talk about that a little bit more later. We're manually annotating the immune response genes. We're doing population variation data across some 1,800 horses mapped to the telomere-to-telomere assembly. And in a paper that is currently in revision, where Eleonora Capoletti and Elena Giolato, who's also on the call here, they worked with me to do the centromere annotation of this, and Carrie Desai down in Adelaide worked with me to do annotation on the repeat regions. So hopefully this paper is going to be coming out soon. Okay, so what has resulted from all of this stuff that has happened? And I would hope that everybody would now agree that we are in the post-genome era, right? If you need one, you can generate it. And currently there's, a proliferation of very high quality genomes. The thoroughbred T is one that was published back in 2004, along with the donkey T assembly. Both of those are available and are annotated. Have gene annotation provided by NCBI. Ensemble is currently working on it. Also, there's a project that I was able to participate on, the ruminant T project. That is in full force now, and you should look at this article if for no other reason. There is this glorious phylogenetic tree that was rendered by Rachel O'Neill for everything in the ruminant clade, and the people who are working on that project are working to produce telomere-to-telomere assemblies for everything in that phylogenetic tree. Some 130 animals, I believe I remember. Okay, so genomes produced in my lab. We've got equine and equid, the ECCAB3 assembly, the thoroughbred T assembly, the donkey T. We currently have an Arabian chromosome Y that we're working to annotate right now, that we got by doing T assembly on an F1 cross of an Arabian sire and a Shire dam, and those will both hopefully be available soon. We've got a female Grevy zebra haplotype also coming soon. We have yet another thoroughbred female haplotype that we're going to be able to add the Arabian Y to. We've crossed a Haflinger dam with a Persian onager. Wouldn't that have been an interesting looking animal? We're working to generate the ONT data for that right now. The HiFi data and the IsoSeq data have been generated for it. And I've spoken with Elena Giolato, and there's a male Zonky out there that would be useful for us to sequence as well. It has a Berkeley's zebra sire, so we could get a zebra Y chromosome. Work that we've done in rat, which of course are not equids, but hey, they're a fun species to work with anyway. The GRCR8, the current reference genome for the rat, was one we generated, an A3 assembly, and we're soon to publish an A3 T assembly, spontaneously hypertensive B2, Wistar Kyoto, F344, Leon normotensive, DAL SR, Leon hypertensive, DAL SS, and LESTM are all genomes that have been completed. These are contig level assemblies, but for the A3 T that has been generated, and a colleague of mine, Dr. Peter Doris, Dr. Melissa Laird-Smith and I are hoping to be able to get the funding to push all of those to T2T status. Okay, pangenome. So these in the horses, this is a project that is being driven right now by Dr. Jonah Cullen and Dr. Sam Stroop. Dr. Cullen is at the University of Minnesota. Sam Stroop is at A&M. Right, so linear genomes are great, but they're obviously not all that. So we know that there's a lot of structural variation that horses have that isn't going to be represented in any one genome, and is going to be difficult to characterize if you're using only one genome, right? This is reference bias, and what we hope to do is remove it. How do we remove it? We, if we do this carefully, hopefully we will enumerate all common haplotypes that would be found in the horse, right? This is ultimately the goal. In so doing, can we identify all common structural variation for the horse? And based on the genomic content, could we identify what haplotypes any horse has? Both of them, maternal and paternal. Can we walk through the pangenome to do that? So some of you out there might be asking the question, well, I'm building a pangenome. How many horses are enough? When do I know I'm done? So I emailed Jonah earlier today asking him for this figure, and he suggested that, well, hey, that's a good question. How do we know we're done? This is of course a good measure of that. So this graph, what this tells you is what new genomic content you have added for every genome that you add. Right. So what you can see here is that this light green, these big bars down here are coverage that's greater than equal to 100%. So this is places where they all agree, and that's going to fall away as you add more horses. And then these other bits, the red are the more rare things, and the gold and the green are the less rare things. But you can see that it kind of sort of plateaus, right? If you keep adding horses, you don't add that much new material, and that's good, right? So are we ever going to get it all? Okay, probably not. That's not a very sensible goal. Are you going to get nearly all of it? And I think that the work that they're doing and all of the horses that they have been able to add into their pangenome, I think that the answer to that is yes. Okay, so a linear genome, right? It's essential, but it's insufficient. It served us very well to broadly understand genomic organization for the interpretation of small variations. Well, yeah. So, single nucleotide variants, small insertions and deletions, we can see where they map on the genome. We can see where they map relative to genes. We can see what, how they might impact an amino acid change or some other such thing. Telomere-to-telomere genomes, they're fully phased and very accurate. They can be nearly complete. So we've advanced this. It's important to remember biology works on the haploid genome, right? The way the maternal chromosomes function is different from the way the paternal chromosomes function, right? And it's important to understand how each of those is manifest. And if you're mapping everything to a single genome, these are things that can be difficult to resolve. So reference bias, simply stated, if it's not in the reference genome, you can't measure it. And here is, I think, a pretty good example of that. This is a figure that was generated by my colleague, Dr. Peter Doris. It is data for the rat. And so there is this protein, ST6-GalNAc1. If we look at RNA-seq data, this is iso-seq data that is generated from the GRC-R8 animal, which was a brown Norway. We see more or less what you would expect. The reads just sort of lining up here. over this annotated locus, all's well, all's good. But then you go to the spontaneously hypertensive rat that's stroke prone, and you look at the same isoseq data, and boy, there's a lot more of it. Well, okay, you stroke your chin and you say, well, maybe it's just that there's a lot more of that particular transcript that's produced by this animal. Could be. But then if you map these same data to the reference genome that we built for that spontaneously hypertensive rat that produced these isoseq reads, you see a more interesting and more believable story. In that spontaneously hypertensive rat, that gene had been duplicated twice. So there are 3 copies of that gene in that rat. And when you map the isoseq data back to that assembly for that rat, you see that each annotated transcript site is covered with reads as you would expect it to be. And you map the GRCR8 isoseq data, and it maps back to the same home that it was able to find in its own respective chromosome. So, pretty interesting story. Obviously, I think that that each of these genomes for each of these breeds, for each of these strains, has something very interesting to tell us. So, why create a pangenome? Genome-wide association studies, they've always relied on an allele being linked to the causative haplotype, whether it's a single nucleotide variant or an indel or an SV. Okay, that's all okay for finding an association, but it's grossly insufficient for understanding the structural basis of a condition. Right? If the reference genome that you are using is not allowing you to articulate the structural variation that is in this animal, you are not going to have a good understanding of what's going on. I think that in the conjunction of the pangenome, low-pass sequencing and SNP chips will likely continue to be used. Why? We'll talk a little bit more about that in a second. So what's produced in a pangenome? Oh, these beautiful pictures, and these were provided to me by Jonah. So here we have we have a node, we have a variation here, and then we've got this variation, but this variant, it has variants on top of it. Who knew? Well, of course that can happen. But these were things that were simply invisible to us before we could do things like this, right? And looking at a linear representation, a more linear representation of it, you see that each of those haplotypes is laid out, essentially showing which of the haplotypes group together, which have variations that allow them to sort. And all the cool structural the insertions, the large insertions, the large deletions, and inversions and whatnot that make genomes so interesting. So identifying variation in a pangenome. Take comfort. So you generate a pangenome, and what's produced, right? What's produced is our old friend, the variant call format file. Isn't this comforting? It's a warm embrace. So if we look at this, we see exactly what we're used to seeing from a VCF file. We see a chromosome, we see a position. So this was built from this VCF file was derived from a progressive pangenome that was built against an anchor that was a T-to-T assembly. This was built for rats. But we've got the chromosome, the position, statistics that we're used to looking at in the VCF files that we see in cohort studies, and then we see our genotypes. Now you'll notice that it's not something / something. It's just a number where the 0 represents the reference allele and the 1 represents the alternate allele. And we can see here that at this position, all of you have this this brown Norway rat, which is the R8. It has a non-reference allele. And here nearly everybody else has the non-reference allele, but the brown Norway and the WKY, and so yeah, you can see what these animals have going on in their individual genomes against the reference file, just the way that we've always used it. That's great. But sometimes a VCF file can look like this too, right? It will, so in this VCF file, you will see articulated everything that's in there, right? This is, in fact, is like a 6 KB insertion. It's a line element that got itself inserted on this chromosome for one of the rats. But it's all going to be there, right? But isn't that cool, right? Previously, you would have been looking at this and you said, I know there's something there, but I don't know what it is, right? And you know exactly what it is, right? So I do the simple thing and I say, okay, I'm going to go through and everything. I just want to know, what are the lengths of those features that are either going into or being taken out of my reference genome. And I made this little histogram, and we see sort of the thing that we would expect to see, where most of the structural variations, or the great big huge peak at 1 for all the single nucleotide variants, but then it kind of decays here, and you get this peak, right, that shows up where full length line insertions ought to show up. Isn't that cool? And then it sort of peters away. We've got 110 sites relative to the spontaneous hypertensive rat that have measured insertions larger than 100 kb. The longest being 242 kb. We can measure that. Okay, so identifying node-specific alleles and using it to assign haplotypes via low-pass sequencing. I think that this is a really cool domain for future research. So if we look here, we see these are, in red, you see alleles that are specific to the brown Norway, right? The brown Norway is the only one that has these alleles. You can imagine, I can write, or I can build k-mers that are specific to these alleles that show up in the brown Norway, and likewise specific to the alleles that don't, right? And then I can look at a raw data set and I can say, tell me with this data set, what haplotype am I on at these positions, right? It's going to tell me, oh, you're on the brown Norway or you're on one of the other ones, right? I can know that just by doing low-pass sequencing. Pretty cool. Okay, and then yes, by taking that data, I can then immediately go to the nodes that are implicated by those alleles, right? So how does any of this help you? That's the question that we all have to get to, right? We can now know how the horse's genome varies structurally across strain. This is a big win, right? When an association and found an inspection of the pangenome can reveal structural differences that may provide biological insights, and the pangenome can suggest the best linear genome for use for analysis, right? If we do find something interesting, maybe we do want to go back and map it to the linear genome, where we might maybe have more tools to be able to scrutinize it. We will know which genome is closest to the feature that has been identified and is the least likely to produce any bad effects from reference bias. Okay, so I hope you agree with me. We're solidly in the post-genome era. What now? Okay, I think we've done. I spent the last 10 years of my career building reference genomes. I want to spend the next 10 years telling people why we don't need them anymore. Right, so reference-free analysis of genomic data. So we've got the pangenomes, and those are going to, we've got the reference genomes, those are going to still be useful. Pangenomes, those are going to still be useful. But when you think about it, right? So when we sequence that yak, when we sequence that mule, what did we get? We got 2 fully articulated haplotypes. You say, you look at me and you say, well, what can I do with that? Think about it. When at the moment of conception, what's that cell got but 2 fully articulated copies of 2 haplotypes, right? It's got what we got, and it can take that information and it can make an us, right? It can make a human, if those are human chromosomes. It can make a horse. It can make a mouse. Whatever it is, it doesn't know anything about our annotation. It doesn't know anything about our reference genomes or our software. It has all of the information that it needs to be able to fully render one of us. Right. So ultimately, where I would like to see us get, at the culmination of my career, is FASTQ to phenotype. That's F2. And you heard it here first. Okay. So if there's a compelling reason to build a haplotype phase telomere-to-telomere genome, I think we can get. We can do that. That's not a problem. Now I think that we need to turn our attention toward physically based predictions of phenotypes based solely on genotype, epigenetic status, or physical or cellular environment. You look at this fun paper. This was from a couple of years back, out of the J. Craig Venter Institute. So, back in my day when I was being a physical chemist and doing molecular dynamic simulations, we were simulating boxes that had 256 argon atoms in them, right? Okay, now, right, 60,887 soluble protein, 2,200 membrane proteins, 1.3 million lipids, 1.7 million metabolites, 17 million ions, 446 million water beads, 6 billion atoms that we're simulating cells, right? Now, I am not naive to the complexity of this problem. It's huge. It's mind-bogglingly huge. But look at how far we've come, and look at every technological barrier that we have blown away in our past. Right? I saw this this wonderful documentary on the Apollo program, and one of the engineers on it said, right now you have more computing power in your pocket than we had on any of those Apollo missions. And I'm not talking about your cell phone. I'm talking about your key fob. Right? We've got artificial intelligence coming down the line. We've got quantum computing coming down the line. And all of these things are becoming less expensive. I believe that we can begin to ponder questions at the level of the cell in the next decade, and I'm very excited to be hopefully be a part of all of that. Since the first nearly complete genome, we've leapt nearly every physical and technological boundary that has constrained us. Full-length, fully phased chromosomes, they're in our near future. I'm relatively certain of that. We need more comprehensive measurements of cellular environments, genetic, epigenetic, transcriptomic, proteomic, and metabolomic, all things otherwise in all ways biochemical. New computational methods that ingest and analyze these data. AI is going to be, it's not going to be an important component. It's going to be a driving component. And we need to enhance real-time and where does the rubber meet the road? How are we helping the farmer? How are we helping the veterinarian? We need to be able to provide real-time decision support for breeders, farmers, and veterinarians. Let's put sequencers on trucks. Let's get that data up to the cloud. Let's get them the information that they need in real time. So, in addition to all of the wonderful people that I have talked about during this presentation at the University of Kentucky, I work with Professor Jamie McLeod, Professor Ernie Bailey. He's retired, but God love him, he still comes in, and I am grateful for it. The Horse Pangenome Project, Jessica Peterson, Molly McHugh, Sean Durward Ackerst, Brian Davis, Siobhan Laux, Jonah Cullen, and Sam Stroop. While I'm on sabbatical, and I've had the gift of a sabbatical for the last 9 or so months, I've been at EBI. Fergal Martin has given me a place to work there. For funding, I have the Clay Fellowship to support my sabbatical. And at the University of Kentucky Department of Veterinary Science, I have the Janet Kohler Endowed Chair of Equine Bioinformatics, which I am very grateful for. I have funding from USDA NIFA and from the NIH for the rat work. Thank you very much. I'm happy to take your questions, but before I do, I want to plug the Havermeyer meeting, which will be hosted in Lexington, Kentucky from August 16th to August 19th. If horse genetics and genomics is your thing, we would love it if you could apply to come to that meeting. We're accepting abstracts now. Please go to the website. Thank you so very much for your time.
Thank you so much, Ted, for this very inspiring and comprehensive overview of pangenomes and also for the future research direction. I will now open the floor for the question. So if you have any, please feel free to unmute yourself or and ask directly or write them down in the chat. And I will be happy to read them out loud. Do we have? Oh, yeah, we have one from Tim Downing. If you want to ask it, unmute you.
Sure, I will, and thanks for a really great talk. It's very, very interesting. And this is a question may have no clear answer, but what metrics and would you use for to quantify this reduction in reference bias if we're using pangenome graphs? Because it's a bit of a diffuse thing. That obviously they're better, but how do we quantify that is tricky. And then this also links to a related question I'm about to put in the chat of what samples. How broad do we need to think? How much diversity should be in a pangenome graph? Because unless we have infinite samples in a pangenome graph, it is always biased in some way. And therefore, we should think about the reduction in that reference bias, because even if we have a really great pangenome graph, it still can be biased. And how do you measure that bias is something I'm interested in. And apologies for a potentially vague pair of questions.
Okay, so I'll take a shot at it. But Jonah, are you there? Are you still there? Okay,
yep, I'm here.
So okay, would you like to take the first bite at that apple?
Yeah, I mean, I'll take the first bite. There's a number of ways to do it. To do that, there's some papers that show like you could just compare your graph-based alignments to the linear alignments, right? You can align it to the backbone from the graph, and then you can calculate various statistics like perfect maps or paired reads, just the proportion of those between your short read data sets against those two backgrounds. There's also tools to do this, like a newer one from last year is called Bias Tools. It's a really nice tool. It gives you some regions where there is some bias based on the comparison to the graph and the linear reference. Yeah, that's sort of the primary ways we do it, or that we've done it so far. I think once you have like a genome in a bottle type, variance set, then you can actually start like assessing that reference bias as it relates to the fraction of alternate alleles versus the homozygous reference alleles. But we don't really have that resource in the horse. So some of the tools allow you to like simulate reads and then do those comparisons as well. But yeah, that's sort of the way that we've been approaching it from the horse space.
Yeah, and I mean, I just follow. I guess sort of end by saying, you got to drive the line somewhere, and you're always going to get that wrong. But if you're studying if you're studying draft horses, make sure you go deep in draft horses. If you're studying speed horses, make sure you go deep in speed horses. So for the problem that you are hoping to understand, endeavor to cover your bases as well as you can.
Thanks.
Yes, sir.
Thank you so much, Theodore. Cedric has a question.
Yes, sir.
Amazing talk. Thanks a lot, Ted. This was
My
really
pleasure.
great. We had Brenda Murdoch a couple of talks ago, and she was talking about the construction of pangenomes. And I don't know much the topic, but apparently there are alternative recipes, and they give quite different numbers, which somehow was vaguely reminiscent of when, in the days when you were assembling your genomes, collapsing repeats or not, and, having long and short assemblies. So what is your take on the current status of pangenomes? And Is this going to crystallize one way or another, or are there just different flavors for different analyses?
Yeah, I'd lean toward different flavors. I mean, all I'm, as you're building these things out, right? I mean, you're even, when we're assembling genomes now, we're not using a pipeline. We're using pipelines, and we're looking to see what seems to give the best result. So the greatest contiguity, the greatest accuracy. Then we move from that place forward. I believe that it's going to be the same with the pangenomes. Figuring out how we write the software that understands this one is the most complete. This is the way that we need to go with this group of animals. That's going to be a challenge, and I think that that's the way we'll need to move forward.
That's a great answer, and sorry for putting on my NF-core hat. So we've been working a lot lately on having pipelines that, rather than applying one magic method to your data, are making available all of the alternative methods and providing a way to combine all of these outputs into one. And so is it something that you think could be the way to go for pangenome, or generally for genome assembly?
Yeah, that certainly does sound a good way to move forward.
Okay, and just out of curiosity, I don't think you've been using much of Nextflow or NF-core, no, on all of this work. Okay, it's okay.
Yeah. So I hang around with the snake crowd, and all good people, all good people.
Yes. It's one thing we said a lot when we were doing Bovreg, this is a cash-strapped community. So if you have stuff working in snake-make, you don't gonna redo everything. But there are good reasons to use an F-core, and I hope we'll manage to convince you as time goes by.
Very good.
Okay, we have another question in the chat from Sean McEachern. I hope I pronounced it right. Who is asking, do you recommend only T2T genomes when building a pangenome, or does short-read assemblies and low-pass genomes contribute much to the project?
So, so we're working, and the horse we're working to answer that right now. So the genome that has been built by Jonah and by Sam, that has a T2T anchor, and everything else in it is contig level assemblies. And that was, so A, that was the data that we had, and B, it seemed that would be a good way to proceed, given that there could have been a lot of errors going from contig level assembly to T2T assembly, right? I mean, we really haven't quantified that nearly as well as we should have, or we should. Another effort that I have going, we're building T2T genomes for 13 breeds of horses, and we are going to build a pangenome with those. And then we are going to ask the question, all right, do you learn more here or there, right? I mean, do you exacerbate assembly artifacts by going all the way to T2T, or is having the centromeres fully articulated, and do you have to mask those? Are those still too big of a problem for these pangenome builders? We don't know. We're going to ask that question, and and low pass sequencing. Low pass sequencing, it remains an enigma to me. I mean, it's always going to cost less. It's always going to cost less, and maybe an order of magnitude less, or more or less. And so for that reason, I don't think it's ever going to go away. But I mean, the fact that using technologies like element sequencing, even Illumina sequencing, the price keeps dropping, right? I mean, at what point does do you you're doing a study, do you say I could do low pass? If it's probably going to answer all my questions, but at the same time, it's not that much more work to generate the 20x data. And do I do that? I mean, I think I believe I honestly believe that SNP arrays are going to be supplanted by low pass sequencing across the board at some point. And it's just incumbent upon us to continue to figure out how to make the most use of them.
Hey, thank you so much again. I have a provocative question, I think, because I wanted to ask, to what extent do you think that reference bias has shaped our understanding of genetic architecture in breeds that are highly divergent, such as some isolated local cattle population? And in light of this, I don't know how to say it. Are we at risk or of be overconfident to inference or systematic underestimation of genetic variation in those population or groups?
I think that our genomes are good enough that assessing population structure using single nucleotide variation is still quite good. But there's absolutely stuff that we will have missed by using only reference genomes. I mean, forever, I remember one of the first studies that I saw that said, hey, the Han Chinese the Han Chinese have 5 megabases of genomic sequence that we were able to assemble out of short read data that weren't in the reference genome, right? Well, those could be an important 5 megabases. And I mean, certainly when you're sequencing to look for rare disease, to try to understand rare diseases, there's a lot of wonky stuff that could be happening that may not be characterized well with a linear reference genome. And in all fairness, pangenomes are not the answer to everything, right? I mean, we're still going to miss stuff. Yeah. Yes, sir.
Francesca, can I ask one more question? So I love this answer you just gave. And one of the things I find amazing, we were talking about the human genome, what did it make possible? And one thing we easily forget is that before the human genome, we had 150,000 protein coding genes, and everybody was selling them. And the human genome told us, it's only 20,000. And just, bounding reality and bounding fantasy is already something useful. Now, I'd like to close this on a slightly philosophical question. You talked about simulations and you talked about understanding things through simulation. Now, if one thing is coming increasingly clear to my mind is that this is all about emerging properties, and emerging properties require a different kind of understanding. If you look at a dead animal or an animal that is alive, or recently deceased animal, it's the same thing. All the cells are still the same. Gene regulation is still roughly the same, as shown by our colleague Roderick Guigo some time ago in GTEx. But one is dead and the other is alive. And so that's clearly an emerging property, which requires some kind of physics. It's probably the same happening with LLM. These are very, very simple mathematical inference, but you do zillions of them and things emerge. Now, when it comes to the pathway to phenotype, which I find fascinating, where shall we look? Because if we look too deep, we will have an excruciatingly neat understanding of the cell, its DNA, but we won't understand the animal. If we look too high, it's what people have been doing forever. It's boring. So what is the right level for looking at phenotypes?
Yeah. So, I mean, I'd be happy for start if you could figure out it was a horse, right? I mean, yeah. I mean, so I mean, I know how to answer that question, right? I would take all the data and I would compare it to all these genes. I know how to answer that question, but we don't have a tool that you could feed this data to, and it will tell you what species it is. right? And then what if and what if I trained that model and then I fed it a mule, right? What would it tell me?
Yeah.
And I mean, so you know, I think there are easy questions that we can answer. And you know, in the world of simulation, I mean, we did that too, right? I mean, we started with simple harmonic oscillators, and we moved to Leonard Jones, and you know, and you know, as we proceeded, more interesting questions became accessible, and yeah, I mean,
very neat. It's a very neat suggestion, because the question is, how deep can you go while still recognizing this thing as a horse, right?
yeah,
Because clearly, if you look at carbon atoms, they have
Yeah.
this thing horse-like. So how deep can you go while still knowing it's a horse?
Yeah.
Very nice, very neat. So I think we're going to have to close, because we've already passed the hour. We've had a fantastic turnout. At the peak, I counted 61 people. I don't know if you counted more people, Francesca, but I think this is by
61,
far
yeah.
the largest turnout we've had. Thanks a lot, Ted, for drawing crowds to our channel today. And Francesca, you may want to pitch our next speaker next month.
Yeah. So in the next talk, we will move from genome completeness to functional resolution, because our next speaker will be Dr. I'm sorry, I will for sure pronounce it wrong. Junjuan Wang, who is a postdoc at the North Carolina State University in Professor Malteca group. And he will discuss fine mapping methods in farm animals. So we will see in the next talk. Thank you so much all for joining, and have a good afternoon.
Thank you.
Thank you all.
Bye-bye.